Hello friends, Ace Botane next episode. In this episode, we'll be discussing biological classification 7th part. And what all things we'll be discussing in this episode? First, we'll be discussing that like a introduction, then subject mainly dealing with the viruses, subviral agents, then as usual, 5 theory question, 5 NEET model question with the key. So we can proceed. Biology classification, 7th part. I will share the screen, so it will be easy for you to observe the photographs, observe the diagrams, and observe the note. It will be easy for you. I think screen came, so we can proceed. Biology classification, 7th part. So when we come to introduction, see, in this episode, certain groups which is not given by Whittaker's classification, lichens we already discussed, other few cellular organization will be discussed, which include viruses, subviral agents, etc. Now we can start with the virus, and this episode is an important one for the exam point of view. Important one for the exam point of view. So we can proceed. Virus. So the term virus was given by M.W. Bejrenick, 1898. That is given in NCERT page number 20, reprinted edition, March 2024. Even though Pasteur's name also suggested, but NCERT clearly says it is M.W. Bejrenick, 1898. And you can find 4 important scientists concerned with the virus. This area I told it is important for NEET exam and theory exam. Theoretical only. So first we can start with Louis Pasteur. Pasteur used the term virus in his work, but he didn't define it in the way it is used today. According to him, it is venom or poisonous fluid. It is in 1880. 1880. That is regarding Louis Pasteur. Now comes second scientist, D.J. Ivanovsky. After 2 more years, that is, not 2 more, 12 more years, 1892. Pasteur 1880, Ivanovsky 1892, recognized in connection with mosaicism in tobacco. And we are able to identify this organism, virus, is smaller than bacteria, able to pass through bacteria-proof filters. That is regarding Ivanovsky's work. Now comes M.W. Bejrenick, 1898, after 6 more years, shown that infected plants can spread disease. And he used a statement, contagium vivum fluidum. We'll be remembering in cell, homini celluli celluli, likewise, contagium vivum fluidum. Meaning is infectious living fluid. And the name virus given by this famous scientist, M.W. Bejrenick. That is what NCERT says. Now comes fourth scientist, W.M. Stanley. Now came into 1935, after many years, 1898 to 1935, almost 37 years. 1935, Stanley crystallized TMV, crystal shown protein nature. So that is regarding 4 important scientists in virology connected with the virus and the contributions given by them. I think idea clear. Usually a question will be there. And remember, NCERT is the base. Otherwise, confusions can come. So NCERT is the base. Now we can proceed to second page. I think first page is clear. Second page. So when we start with second page, viruses not normally seen in general classification. That is why in Whittaker's classification, we are not able to identify it, or is not able to identify it. Many reasons, including they are not living outside a living cell or live cell. So they can live only in a live cell. They can show life property. They lack a cell. So that is why acellular. Many consider that viruses are connecting between living and non-living. This property will be comparing in coming pages. Living nature and non-living nature of virus. And remember, viruses are obligate parasite important. Obligate, strict parasites. Now coming to structure of virus. Viruses got a simple structure. Only 3 components will be normally present. 3 components. Nucleoid and capsid. These 2 are must. Envelope may be seen, may not be seen. So virion, another term, virion, an inert virus outside of a host cell. Otherwise, other definition, a complete infectious form outside. That is virion. Now comes first component, nucleoid. Nucleoid means genetic material. It is the genetic material. Viruses have DNA or RNA as the genetic material. In DNA and RNA, double-stranded and single-stranded nature can be seen. That means double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, another important area of question. Now, double-stranded DNA and single-stranded DNA, so everything given there will be easy for you. Double-stranded DNA and single-stranded DNA, many examples are there, specifically double-stranded DNA, single-stranded DNA comparatively less. So example for double-stranded DNA, herpes virus, pox virus, cauliflower virus, mosaic virus, etc. Single-stranded DNA, it is comparatively less. That is 5x174, it is a coliphage. I think idea clear. Now, double-stranded RNA, wound tumor virus, reovirus. Single-stranded RNA, tobacco mosaic virus, FNM means foot and mouth disease virus, retrovirus, influenza virus, etc. Now, after that, again, an important item, plant viruses, generally single-stranded RNA. Animal viruses, generally double-stranded DNA or single-stranded or double-stranded RNA. So, DNA will be double-stranded. It can be single-stranded or double-stranded RNA. Now, bacteriophages, frequent question. That is bacterial viruses. They have double-stranded DNA. So, that part is an important area for theory and runs. I think page is clear. Now, we can proceed to next page. See, normally the word phage is coming as a suffix. So, example, bacteriophage, polyphage, cyanophage, mycophage, zoophage, phytophage, etc. So, suffix phage, prefix that organism affecting will be shown. So, example, bacteriophage means viruses which infect bacteria. Coliphage means viruses which infect Escherichia coli. Mycophage, viruses which infect fungus. Phytophage, viruses which infect plants. So, prefix generally indicates the organism affected by respective phages. I think idea clear. Now comes capsid. 1 mark theory question even. It is normally outer covering made by protein. Its basic unit is called capsomeres. Protein in nature, and it is basically for protecting the genetic material, that is capsid. Protein, capsomeres, protection. Now, third component, that is envelope. See, this structure is not seen in all viruses. We cannot find in all viruses. It is thin and loose. Thin and loose, chemically composed of proteins. From where that protein comes? From virus. Also, lipids will be there. From where that lipid comes? Host. So, area of question, protein from where, lipids from where. Typical example is herpes virus and HIV virus. Now, all envelope viruses have capsid. Capsid will be there. And after capsid only envelope comes. So, if envelope is there, capsid will be there. But envelope is not seen in all viruses. Capsid is present in all viruses. So, I think that line is clear. Now, structure of TME and bacteriophage, two important diagram from NCERT, figure number 2.6, labeling, explanation of all this. Secondary also it comes. Now, first is tobacco mosaic virus. So, before getting into TME, I told you we'll be discussing living and non-living nature of virus that I given on sides of the TME. So, living nature of virus. Many things are there to explain its living nature only. 4, 5 I given there. DNA or RNA is a genetic material. So, presence of genetic material. Multiplication ability, even though it is in host. Presence of mutation, variability. That is why we are familiar with the coronavirus. Specific to host. These all are few items explaining living nature of virus. Now, what about non-living nature of virus? Able to crystallize, showing it is a chemical. Not showing life outside a cell. No protoplast, no respiration. High specific gravity. High specific gravity normally belongs to non-living objects. So, comparison or a few difference between living and non-living nature of virus. Now, general features of TMB. Famous diagram. Size and shape, around 3000 Armstrong in length, 180 Armstrong in diameter. Molecular weight roughly 39.4 into 10 raised to 6 Daltons. Capsid is made by capsomeres. It is 2130. Diagram we can find is here. Arrangement is helical. See, helical. And genetic material, single-strand RNA, here we can find. Single-strand RNA, again helical. Consists of 6400 nucleotides. Thus, the ratio between capsomeres and nucleotides. Capsomeres 2130, nucleotide 6400, roughly 1:3. Famous question. I think page is clear. Now we can proceed to next one, TME. Now comes bacteriophage. TME is over. Tobacco mosaic virus is over, diagram over. Now we are proceeding with the bacteriophage. See, these are viruses which infect bacteria and they possess double-stranded DNA. When we look onto the bacteriophage diagram according to NCERT, T4 resemble, this T4 resemble shape of a tadpole. Tadpole means head will be there, tail will be there, connecting area will be there. That's the reason. It has got a head here. In bacteriophage, it has got a head. Polyhedral shape you can find. Here the shape. Polyhedral attached to tail. See here, you can find tail, which is helical in nature. Now, nucleic acid is in head. Is double-stranded DNA, so important item, bacteriophage. Shape of the tail is tube-like, chemically proteinaceous. One end of the tube is joined to the head by thin collar area. Here you can find collar. The other end, a plate, external in shape. And the plate has got 6 small tail pins and 6 tail fibers. Number is important. Function is helps to attach with the host cell. Helps to attach with the host cell. That is function of tail pins and fibers. Now comes reproduction virus. So when we come to reproduction virus, basically 2 types, phagic type and pinocytic type. Phagic type, that is again divided into 2, lytic cycle and lysogenic cycle. So what is meant by phagic reproduction? In phagic reproduction, only nucleic acid of the virus enters into the host cell. So in phagic type, only nucleic acid of the virus enters the host cell. It is 2 types, lytic cycle and lysogenic cycle. So what is lytic cycle? It is seen in virulent phages, bacteriophages T4, and lysogenic cycle. It is seen in temperate virus. Temperate virus normally means non-virulent. Immediate cell death may not take place. Example is lambda phage. I think up to that it is clear. Now we can continue with the next page. Pinocytic cycle. So phagocytic and pinocytic, or phagic and pinocytic. In pinocytic type, here the entire virus enters into the host cell, except only envelope. Envelope will not be entering if it is present. Otherwise, entire virus may enter into host cell. That is pinocytic type. Now comes some viral diseases. Many are there. Typical examples are given: plants, animals, and humans. So when we come to plants, animals, and humans, plants, generally viral disease commonly called mosaicism. That is cauliflower mosaic virus, tobacco mosaic virus, likewise. Then leaf roll, potato, example. Bungy top, typical example is banana. So famous disease, bungy top disease of plantain, banana. Leaf tail in papaya. Now animals, FNM. FNM means foot and mouth disease. In cattle, bird flu, avian flu, we are familiar. Panic in fowls. Now humans, smallpox, chickenpox, common cold, yellow fever, mumps, measles, and encephalitis. Then SARS, COVID, coronavirus, all familiar. Now, architectural forms of virus. Some point I little bit gone over NCERT because referring previous year question, few question came. That is why little bit extra I given there. Basic book is NCERT, no doubt. Architectural forms of virus. 3 forms it can exist: helical form, cuboidal form, binal form. Helical means elongated body, as in the case of TMB. Here we can find tobacco mosaic, helical form, elongated body. Now, cuboidal, that is not here. That is poliovirus. Then binal. Binal is 1 plus 2. That is helical and cuboidal together, binal. Example is 2 phage. I think idea clear. Now, subviral agents. They are indirectly viruses, not original virus. Indirectly, we can say indirectly, we can only use the term virus. Indirectly viruses, but lacking some essential component. 3 groups are discussed. Basically, 3 groups will be discussed. Component, I think one spelling is wrong. COMPONENT. One N is extra. Sorry. So component. One N is extra. Sorry. 3 groups will be discussed: prions, viroids, virusoids. Now, prions. Prions are proteinaceous infectious particles, PRP. They are connected with the spreading of certain infectious neurological diseases. And they have folded proteins. Virus and prions have almost similar size. Virus and prions have almost a similar size, almost. Now, some important diseases caused by prions: CJD, Creutzfeldt-Jakob disease, mad cow, BSE, bovine spongiform encephalopathy, Kuru, laughing death disease in humans, scrappy in shapes. Now, Stanley Procina, connected with the term prions, and Tikava Alper, crucial contributions in prions. They established that is proteinaceous. Now comes viroids. So when we come to viroids, meaning of the term, virus means poison, eidos means diminutive, small. And viroids are the smallest self-replicating particles. Viroids. Now, discovery by Diner in 1971, RNA particle, infectious, no protein coat. And they are obligate parasite, molecular weight is low. So viroid is important for NEET exam. Molecular weight is low. And viroids are causing diseases at least 20 in plants. Animal and human infections not yet known. Viroids are smaller than virus. Some diseases caused in plants, potato spindle tuber disease, PSTD, chrysanthemum stun, citrus exocortis. Now comes next component, virusoids. Scientist is Randley and et al. Genetic material is RNA. They are RNA viruses, but seen inside the capsid of large virus. Replication the host, and they will not cause any infection. That is what up to today science know normally. They will not cause any infection. That is why I given the term at present. Later, we may able to observe it. Then after that, 5 theory question, 5 NEET model question with the key. So just to summarize what all things we learned in this episode. Bioclassification, 7th one. Mainly, we are starting with the viruses. Cell organization will be discussed, not explained in Whittaker's system. Lichens already done. Now when we come to virus and subviral agents. Now virus, 3 important 4 scientists, their contribution, very important for exam point of view. Then where we can find viruses. Then a cell organization, and viruses are obligate parasite. They are supposed to be linked between living and non-living. Structure of virus, 3 components, nucleoid, capsid, and envelope. Nucleoid is genetic material, can be double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA. Examples will be important. And bacteriophages, it is double-stranded DNA, frequent question for NEET exam. Bacteriophages. Then what is meant by phage? What is meant by the term phage comes as a suffix like cyanophage, mycophage, etc. Then we entered into capsid, that is outer covering, protein, basic unit capsomeres, protection. Then envelope, not in all viruses, thin and loose, chemically composed of proteins from virus, envelope. Then lipids from host, example HIV, herpes virus, etc. All envelope virus have capsid. Then structure of 2 viruses, NCERT figure number 26. Then tobacco mosaic virus, TMV. Now we compare living and non-living nature. Here and here we can find comparison. Then general features of TMV, their shape, size, weight, molecular weight, then capsid, how many capsomeres, how many nucleotides, ratio, single-strand RNA, helical, etc. Then bacteriophage, NCERT diagram again, double-stranded DNA, bacteriophage infect bacteria. Then diagram of T4, bacteriophage, like a tadpole larvae. Then polyhedral head, tail, then double-stranded DNA. Then there will be polar area. Then bottom side we can find 6 small tail pins and 6 tail fibers help to attach. Attach, I think E is not needed. Attach. Then basically reproduction 2 types, phagic and phagocytic. Phagic, lytic and lysogenic cycle. So what is meant by lytic cycle and lysogenic cycle? Then next it is phagocytic type. Entire virus enters into host cell except only envelope. Previous case that is only nucleic acid enters. Then some viral diseases to plants, animals, and humans. Then 3 architectural forms of virus, helical, cuboidal, binary with example. Then subviral agents like prions, viroids, virusoid, proteinaceous particle. They have folded proteins. Virus size and prion size almost similar size. Then some important disease caused by prions. Then Stanley Prusiner and Tikava Alper. Stanley Prusiner with the name prion, Tikava Alper, crucial contributions including protein establishment. Then viroids. Viroids meaning poison, eidos means diminutive, smallest self-replicating particle, discovery by Diner in 1971, no protein coat, RNA particle, infectious, obligate parasite, molecular weight is low. It is causing, viroids are causing 20 diseases at least in plants, animal and human infections not yet known. Viroids are smaller than virus. And some important diseases like PSTD, chrysanthemum stun, citrus, exocortis. Then virusoids, Scientists run DALI. Genetic material is RNA. RNA viruses, what's inside the capsule of large virus. Replication in the host, they will not cause any infection at present. Then, after that, 5 theory question, 5 NEET model question, as usual with the key. If you like all this episode, subscribe all, like it, share it, comment on it. We will remember daily. Now 2 questions I'm putting as short. One is from second year and one is from first year. One will be around 12:30 or 12 to 2 time after lunch, etc. Then other will be after 8:30 or 9. That way it will be putting 2 shorts, one from first year, one from second year. So time being we can wind up. Thank you. Take care. Bye.